Good morning or afternoon. I am Dr. Meiliyana. On this occasion, the topic I will bring is the introduction of parasitology.
The following is a table of contents that will be discussed. Starting from definition, taxonomy, and classification of parasites and fungi. Hosts and parasites. Transmission. Life cycle, in general. Pathogenesis. Laboratory diagnosis of parasitic and fungal diseases, at a glance. Immunoparasitology. First, definition. Parasitology is the scientific discipline concerned with the study of parasites, which will be discussed in particular regarding human or medical parasitology. The word parasite comes from a Greek word meaning one who eats at someone else's table. Biological meaning is an organism that lives on or in a host organism and obtains its food from or at the host's expense. The scope of parasites discussed are helminths, protozoa, arthropods, and fungi. Moreover, this is the taxonomy, with the top one being the superkingdom and the bottom being the eukaryotes. Helminths and arthropods belong to the animal kingdom, protozoa and fungi belong to a separate kingdom. The history of the discovery of parasitic organisms that can cause infection in humans was first reported in 3000 to 400 BC, especially in 1500 BC found writings on the Ebers papyrus scrolls about parasitic infection by intestinal roundworms and tapeworms in humans in ancient Egyptian cities. After that, it was also reported the discovery of parasitic infections in Greece, China, India in the century BC. Then in modern times, around the 1500s, several important figures emerged in the discovery of parasitic infections known to this day, such as Francesco Redi, father of modern parasitology who has illustrated more than 100 parasites, C.V.A. Lavrin important findings regarding malaria infection and Patrick Manson, the discoverer of filariasis. Parasites, based on their habitat in the human can be classified into ectoparasites are parasites that live attached to the outer surface of the host's body, for example arthropods. While endoparasites are those that live inside the host's body. Invasion by endoparasites is called infection. For example, helminths which are multicellular, and protozoa which are unicellular. Endoparasites based on the necessity of living in the host's body are subdivided into obligate, facultative, parasitic or free-living, incidental or accidental, erratic or aberrant. Endoparasites can be intra or extracellular, and this is important regarding immunoparasites. Fungi can be divided into unicellular yeasts and mold, which is multicellular and has hyphae, mycelia. Another classification is dimorphic, which is discussed in more detail in the KP3 block. Next, the host-parasite interaction. The host is the living organism from which the parasite obtains food and protection. Host-parasite interactions can be commensalism versus parasitism symbiosis. Note, in fungal infections, there are also host interactions, namely commensalism, parasitism, amensalism. The classification of the host can be divided into definitive host, intermediate hosts, reservoir hosts, parademic or transport host, incidental or accidental host. Next is transmission, which is how the pathogen is transferred from host to susceptible host. It can be seen from this circle image that transmission from one host to another can be divided into modes and sources of transmission. In more detail, the distribution will be a task on the day of the class schedule. It is important to know that the transmission source can come from the host's body. The microbiome consists of both helpful and potentially harmful microbes that inhabit the human body. For fungi, modes of infection can be divided into direct contact, for example, tinea or ringworm at cause dermatophyte fungi and inhaled fungal infections that cause Aspergillus. Furthermore, the source of infection can be divided into endogenous colonization by members of the normal flora such as Candida albicans or reactivation of previous infections such as Cryptococcus, and exogenous such as through the air due to Aspergillus or through percutaneous. Transmission of pathogens from the current host to future hosts is a repeating cycle which is commonly known as the parasite life cycle. In helminths and protozoa, the life cycle can be direct or simple and indirect or complex transmission. Direct transmission only needs one definitive host, henceforth, indirect occurs through multiple hosts who are definitive hosts, one or two intermediate hosts. Notes: humans can serve as definitive, intermediary hosts. In arthropods, the life cycle is better known as the division of complete metamorphosis or holomoboly, which includes 4 stages, and incomplete or hemimetabolous metamorphosis includes 3 stages. The life cycle of fungi is better known as reproduction, which can be divided into asexual or vegetative reproduction such as budding in yeast fungi and fragmentation or through asexual spores in multicellular molds. As for sexual reproduction in multicellular molds through sexual spores and in yeast through the mating factor pathway. The principle of pathogenicity of parasitic and fungal infections is related to several things. First, the habitat of pathogens, whether intracellular such as Plasmodium protozoa or extracellular such as helminth infections, arthropods which are later associated with immune response or inflammation, which are discussed in more detail in immunoparasite topic. The second is virulence. The third is immune evasion ability. And the 4 sizes of the pathogen related to whether it can cause trauma or obstruction. And the 5 morphology of the parasite, for example, hookworm's mouth. Methods of laboratory diagnosis of parasitic and fungal diseases can be distinguished by macroscopic and microscopic examination. Culture. Immunodiagnosis like antigen or antibody detection. Molecular detection or PCR. Imaging. Inoculation in animals, which is now rarely used. All the samples for this laboratory methods can be from stool, blood, urine, etc. samples. In addition, according to WHO, some parasitic and fungal infections are included in neglected tropical diseases, as shown in the table. In addition, the Indonesian Ministry of Health has a target to eliminate malaria completely by 2030. Details or further information on this entire introductory material will be an assignment during the lecture session. In the end, thank you for your attention. See you in class.